Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everyone, welcome if you're new, welcome back if you're a repeat listener. Today we're continuing our discussion of metabolism with metabolism of lipids. We're going to talk about buildup, storage, breakdown, and everything in between. The first thing to talk about is the major building block of fatty acids, and really the major breakdown product too. It's all going to come down to acetyl-CoA. All roads lead to acetyl-CoA, all roads kind of lead from acetyl-CoA, and we want to just remember that acetyl-CoA is made primarily in the mitochondrial matrix, which is the same place where fat is going to be oxidized or broken down, but fatty acids are actually synthesized in the cytosol, and that's going to be important for regulation, and we'll come back to that. So what's the energetic value of fat? Why would we make fat? There's actually much more energy storage per unit in a fat molecule than there is in, let's say, a glycerol molecule, because they're more highly reduced, so there's a lot more to be oxidized and to release energy. And also they can be packed very tightly, so even just space-wise, they're extremely efficient, and a person can live off their fat stores for like a month. So energetically, these are actually very valuable. So we're just going to go over some basics about fatty acids before we get started. So first of all, let's give a definition. What is a fatty acid? This is a carboxylic acid with a long hydrocarbon chain, and if you have 3 of them attached to a carbon backbone like glycerol, it becomes a triacylglyceride or triglycerol. Sometimes people will equate and they'll kind of say triacylglyceride and fatty acid interchangeably, and that's not technically correct because fatty acids are the constituents that make up triacylglycerides. Triacylglycerides are made up of 3 fatty acids kind of stuck together, so they're not exactly the same thing even though they're similar. But it's important to differentiate them because we're going to have to talk about how triacylglycerides are broken down into fatty acids in order to be used and how fatty acids are built back up into triacylglycerides for storage, so we just want to get that straight. In terms of the fatty acids that we produce, we only have even-numbered fatty acid chains produced in our bodies because the way our synthesis works is that we add 2 carbon units at a time. We can have odd-numbered chains that we get through our dietary intake, and we can use those, but the ones that we synthesize are only going to be even-numbered chains. I also want to make sure we remember the difference between a saturated fat and an unsaturated fat. So when we say saturated fat, what we mean is that it's saturated or filled with hydrogens. So we know that carbon likes to make 4 bonds, and when carbons are lined up in a linear chain, they by default have 2 bonds, one with the carbon on the right of them and one with the carbon on the left of them, so to speak. And so they have free open bonding spots, and those are taken up by hydrogens. So if you have all single bonds, you're going to have 2 hydrogens bound to every carbon. Now, if you have any double bonds, then it becomes an unsaturated fatty acid because suddenly you don't have 2 hydrogens bound to every carbon because some of the carbons have 3 carbon bonds on them. And in our bodies, we only make cis double bonds, which means that the hydrogens that are left over on the carbons end up on the same side of the bond. We can see trans double bonds, but these are not naturally synthesized. These are created by hydrogenation, so they can be present in synthetically produced foods, but in any naturally produced fatty acids, we're only going to have cis double bonds. I want to take a minute to talk about fatty acid nomenclature. There are 2 or really kind of 3 ways of doing it. So the first way is common names. So certain fatty acids get common names like linoleic acid, palmitic acid, arachidonic acid. And these names mean a certain number of carbons with a certain number of double bonds in certain places. But aside from those, and those names are not very descriptive, we actually have descriptive names that tell you about how the molecule is formed, and we have 2 different ways of doing this. The nomenclature for both of them is dependent on the chain length and the double bond location, but we have omega nomenclature, which counts the double bond location from the free end of the chain, and we have delta nomenclature, which counts the double bond location from the carboxyl end. So for example, palmitoleic acid. We're going to name it both ways just so you can kind of see how this works. So if you give it the delta nomenclature, it's going to be a 16-1 delta 9, which means it has 16 carbons, 1 double bond, 9 bonds away from the carboxyl end. If we give it the omega nomenclature, it's going to be 16-1 omega 7. Again, 16 carbons, 1 double bond, 7 bonds away from the omega end or the free end of the chain. So both tell you that it's 16 carbons, both are telling you how many double bonds and the location of those double bonds, but it just matters which end you're counting from. I also want to mention essential acids. What are essential acids and why do we care about them? So Essential omega acids like linoleic acid and alpha-arachidonic acid are fatty acids that we can't make ourselves, but we make very important unsaturated fatty acids from these essential acids. And the molecules that we make for these are important for building membranes as well as for important signaling molecule precursors like phosphatidylinositol, which, if you remember, is cleaved to pip-2 diacylglycerol in many signaling pathways. Many of our phospholipids have stearic and arachidonic acids, which are essential omega acids, meaning we can't synthesize them ourselves, but we need them. Aside from that, there's another clinical significance to arachidonic acids, just to be familiar with that name, because it's a precursor to many bioactive hormones associated with inflammation. So aspirin, for example, works by inhibiting the production of these arachidonic acid products that we call prostaglandins, which cause a lot of our inflammation. Okay, so let's talk about how we make fatty acids. This is only ever going to be done under high nutrition conditions where we have tons of glucose, TCA cycle is inundated, and more glucose just keeps coming. So the body's like, what do we do with it now? We have to store it. And one way we store it, as we know, is as glycogen. And another way we can do it is that acetyl-CoA can be made into fatty acids and stored in lipoproteins. This is going to occur in the cytosol of the liver, remember, not in the mitochondria. And the process of producing fatty acids is a reduction process. Essentially, it's a reversal of the beta oxidation, which is the breakdown of fatty acids, which we'll talk about a little bit later. Without going through the steps in any detail, because they're really not important for you to know at this point in your career, we use NADPH and we add carbons onto a growing chain, 2 carbons at a time. And even though I told you that it's made from acetyl-CoA, it is, but the acetyl-CoA actually has to be transformed into malonyl-CoA before we can use it, and that's produced by carboxylation of the acetyl-CoA. And this production of malonyl-CoA from acetyl-CoA is the rate-limiting step of fatty acid synthesis, and we'll talk about its regulation soon. And then there's another set of enzymes that do elongation and add double bonds and are involved sort of later down the line, but this first transformation of acetyl-CoA to malonyl-CoA is the rate-limiting step. So how do we get free acetyl-CoA in the cytosol? This is important because remember that acetyl-CoA is formed in the mitochondria by pyruvate dehydrogenase from pyruvate, and we have to somehow get it into the cytosol where all the enzymes, particularly acetyl-CoA carboxylase, that rate-limiting step enzyme, are going to be living. But the only way anything can get in and out of the mitochondria, just to remind you, is by a transporter. The mitochondrial membrane is extremely guarded, and it doesn't just let things in and out freely. So in order for things to get in or out, they need a transporter. And it happens to be that acetyl-CoA does not have a transporter that can take it from inside the mitochondria to out in the cytosol. So what acetyl-CoA has to do is it has to actually enter the TCA cycle, go along until it becomes citrate, and then we can bleed out citrate from the TCA cycle, and citrate has a transporter that can take it out into the cytosol. Once in the cytosol, it has to be converted back to acetyl-CoA. This is done by an enzyme called citrate lyase that forms oxaloacetate and acetyl-CoA, and it also produces NADPH, which is very convenient because we're going to need NADPH very soon as we do this fatty acid synthesis. Now there's a cancer correlate here, and I like to throw it in cancer correlates because they're interesting and they're important and they're relevant. So citrate lyase, like we just said, turns citrate into acetyl-CoA. This is the only way to get acetyl-CoA in the cytosol for de novo fatty acid synthesis, and therefore this is being investigated as a potential drug target which could inhibit tumor growth. Tumors tend to rely very heavily on lipid metabolism and on building their own fatty acids, possibly because they need a lot of membranes for their explosive growth, and the amount of fat that we ingest, which might be enough for a cell at normal metabolism, is not enough for cancer cells, for tumors, which are trying to build all these cells super, super fast. Anyway, just an interesting point. So back to where we were, where we got acetyl-CoA formed in the cytosol from citrate, and now we have to form malonyl-CoA. The enzyme that does this is a carboxylase, and like all carboxylases, what this one does is it adds a carboxyl group with biotin as a cofactor. Biotin is B7 for those of you who've forgotten. So acetyl-CoA carboxylase takes CO2 from bicarb actually, and it swings around and puts it onto acetyl-CoA and forms malonyl-CoA. And then malonyl-CoA undergoes a stepwise process, whereas it loses that carbon actually pretty quickly and gets added to another malonyl-CoA which lost its carbon pretty quickly, and we get this carbon chain being built up 2 carbons at a time. Now how is this process regulated? Because obviously there's always going to be regulation, and like most processes of metabolism, we have 2 levels of regulation. We have the hormonal regulation and we have the allosteric regulation. So the hormonal regulation is fairly intuitive and straightforward. Insulin is going to upregulate this process because remember, insulin is released when there's lots of glucose in the blood, and we want to be in an energy storing or energy using sort of phase. And also, like a lot of other regulation, the way this works is by phosphorylation and dephosphorylation of an enzyme. So insulin is going to activate a phosphatase, which dephosphorylates acetyl-CoA carboxylase, makes it more active so that it can transform acetyl-CoA to malonyl-CoA and allow for fatty acid synthesis. In the fasted state, the opposite is going to happen. Glucagon is going to activate a kinase, which is going to phosphorylate the enzyme, lower its activity, and we're going to do less storage. Allosterically, this process is going to be positively regulated by citrate, which makes sense. In the fed state, you have a lot of citrate, and citrate feeds directly into this process. It's going to be negatively regulated by long-chain fatty acids, which is basically feedback inhibition. So as the long-chain fatty acids build up that we've synthesized, they're going to feed back and prevent the synthesis. So now once we have these free fatty acids, either from the synthesis that we just discussed or from dietary uptake, which we'll discuss in a couple minutes, how do we store them? So typically, free fatty acids don't just live the way they are. We store them as triacylglycerides. And triacylglycerides are a 3-carbon backbone called glycerol, which has 3 long chains on it. And unlike the differentially polar nonpolar phospholipids that we think about when we usually think about coupling a backbone to long fatty acid chains, these are actually fairly neutral and nonpolar all around, which is useful for them. And once we make these triacylglycerides, they can be stored in lipid droplets. And we have lipid droplets in every cell, but primarily the main storage depot of lipid droplets is in adipocytes, which you might imagine. Okay, so let's talk a little bit about the structure of these lipid storage molecules. So lipid droplets and the other kind of lipid storage molecule called lipoproteins have a similar structure. You have a core of fats surrounded by a phospholipid monolayer, which has the acyl chain pointing inward towards the lipid droplet. It's not a micelle, which if you recall, is a structure that forms spontaneously in an aqueous environment to protect the hydrophobic portion of a molecule. This is much more sophisticated than that. It's like an actual particle, whereas a micelle is something kind of temporary that forms spontaneously and can be unformed pretty easily. There are also going to be proteins associated with the monolayer, which can be perched kind of on top of it on the outside or nestled in the layer, but they don't go all the way through. They're not transmembrane proteins. And the point of these proteins, which we will discuss, is that they allow access to lipolytic enzymes which can come in and hydrolyze the triacylglycerol back to fatty acids and glycerol when we need to use it. So how do we make triacylglycerides from free fatty acids? When free fatty acids enter a cell, they almost immediately get conjugated to CoA, forming fatty acyl-CoA, and this traps them inside the cell. This formation of fatty acyl-CoA is so-called activating the fatty acid using ATP to bind the CoA to the acyl in irreversible conversion, forming a thioester bond between the CoA and the acyl. The fatty acyl-CoA, which is now activated, is then added to a glycerol phosphate backbone, and then the CoA is kicked off. Now I said glycerol phosphate backbone, so the glycerol phosphate backbone actually only has 2 free sites. So there is a choice here that either 2 or 3 chains can be added, 3 in the case of a triacylglycerol and 2 in the case of a phospholipid. So what happens is that the first 2 chains get added on, and then there's kind of an option to continue or not. So the phosphate has to be kicked off after the first 2 additions to expose the 3rd site. So they all start out as phospholipids, and then they either stop there or they go on to add 1 more chain. And again, just to reiterate, unlike the membrane lipids, the phospholipids, which stop there, once you add that 3rd chain, the triacylglycerides become nonpolar, and they're actually quite neutral. So we talked about this glycerol backbone. Where do we get the glycerol from? So in both the liver and the adipose tissue, which are the 2 places where we are going to be mainly thinking about making and storing triacylglycerides, even though all cells can do this, glycerol phosphate can be made by reducing DHAP from glycolysis to glycerol phosphate. Or, and this can only be done in the liver, free glycerol from the hydrolysis of a triacylglyceride, which is maybe done in the adipose and then stuff is shipped over, can be directly phosphorylated and recycled to create a new triacylglyceride kind of on the spot. This lipid droplet or lipoprotein formation actually occurs in a very interesting way. So the triacylglycerides, the process of attaching the free fatty acid chains to the glycerol backbone, this all happens in the endoplasmic reticulum. And the triacylglycerides tend to pool or kind of collect between the leaflets of the endoplasmic reticulum membrane, which remember is a phospholipid bilayer. And this pool of triacylglycerides is going to grow and grow until you have a large bulge. You can actually see the membrane bulging out into the cytoplasm. And at this point, you have the droplet mostly surrounded by the cytoplasmic leaflet of the endoplasmic reticulum. And eventually this droplet is going to pinch off, and it steals the endoplasmic reticulum membrane with it. So it just kind of kidnaps this piece of membrane, which it uses to form the coat of its lipoprotein or lipid droplet. And then the perilipin proteins, which are the droplet coating proteins, get attached afterwards. Now we have this lipid droplet floating around in the cell. And how do we do lipolysis of the lipid droplet when we need it? So the triacylglyceride as a unit is not really useful. It's really strictly a storage unit. The whole thing has to be broken down into fatty acid and glycerol, both of which can be used separately if we need to use it. So in the fasted state, glucagon is going to activate cyclic AMP, which frees PKA, protein kinase A, remember, which phosphorylates a lipase called hormone-sensitive lipase, which makes sense because it's responding to a hormone, glucagon, right? Which is then activated, and this hormone-sensitive lipase, what does lipase do? It breaks apart fat, right? Can enter the lipid droplet and break apart the triacylglyceride. The free fatty acids are then going to be released, and they're transported out of the cell, conjugated to albumin, which is a carrier protein, and they're carried to the appropriate cell that needs them, or they go wherever they're needed within that own same cell. Now, when the fatty acids reach the cell that they are intended to go to, they enter by fatty acid transporters. They need transporters because at physiologic pH, fatty acids are going to be charged because their pKa is lower than physiologic pH. So they need transporters to get across the membrane. Once they get across the membrane, the same thing happens to them as we said before. They're immediately conjugated to CoA, but in this state of breakdown, they're going to be taken to the mitochondria for beta oxidation. And there's a little bit of a nuance there, and when we talk about beta oxidation in like 2 minutes, we'll go through that. But first I want to talk about how else can lipid droplets be broken up, because this is not the only way. There's another way. They're also subject to autophagy, so they can be taken up by an autophagosome, which eventually fuses with a lysosome, which will break everything down. And then we also have to talk about lipolysis done outside the cell, because this was, remember, if the droplet is still hanging out inside the cell. So why would we ever want to do lipolysis outside the cell, you might ask? And the answer is, in order to digest fatty acids. Most of the fat that we eat is going to be in the form of triacylglycerides, but those, if you recall, can't get into the cell by themselves. So in order to be absorbed, the lipases of the intestines have to emulsify the fat with bile salts and turn them into free fatty acids, which can then be taken up by intestinal epithelial cells. So the fatty acids are taken into the intestinal cells. They get shipped to the endoplasmic reticulum of the intestinal cell, processed back into triacylglycerides so they can be packaged and sent away. Once they're all packaged up neatly into lipoproteins in the endoplasmic reticulum of the intestinal cell, they're secreted out the basolateral side, and instead of joining the bloodstream and going to the liver where they would undergo a lot of heavy first pass metabolism, instead they actually go to the lymphatics. The lymphatics pick up these lipoproteins, which are called chylomicrons, and then they eventually release them into the bloodstream, but they kind of bypass the liver. So the lipoprotein assembly in the intestines is pretty similar to the lipid droplet lipoprotein assembly that we talked about in any other cell, except that there's a special coat protein called ApoB48, which is used in intestinal cells. This is a truncated form of the protein ApoB100, which we see on lipoproteins produced in the liver. And like I said, these lipoproteins which are formed in the intestines are called chylomicrons. What happens to these chylomicrons? So the chylomicron is born with ApoB48, and then once it reaches the bloodstream, it gets additional proteins on it, ApoE and ApoC2, which are not permanent. They kind of hop on and off as needed. So the ApoC2 activates lipoprotein lipase, which is another lipase, and it acts like the cytoplasmic lipases to release fatty acids to cells. And then the remnant of the chylomicron, which still has like some cholesterol and maybe a little bit of fatty acids left in it, is taken up by the liver, and this uptake is moderated by ApoE. The liver then turns this remnant of a chylomicron into VLDL, very low density lipoprotein, which is structured similarly to chylomicrons except that it carries ApoB100, and then it gets shipped out and it goes through the same lysis process in the blood. ApoC2 turns on lipoprotein lipase and it delivers whatever triacylglycerides it needs to deliver. The remnant goes back to the liver. One thing to note, and one issue that can come up sometimes with lipoprotein lipase, is that it's made under the blood vessels. Actually, it's not made by any cells inside the blood, and it has to be able to get into the bloodstream. The way this works normally is that it gets endocytosed on the basal side of an endothelial cell and then exocytosed out the luminal side into the blood vessel. But if you have a defect in any part of this system, you're going to have a deficiency of lipoprotein lipase in your blood, which will not allow you to clear chylomicrons from the blood. So you may see a phenomenon where normally chylomicrons may give the plasma a sort of milky appearance, and this is cleared pretty quickly by lipoprotein lipase. But if you have a defect in this system and you can't get enough lipoprotein lipase into your blood, you're going to see a very milky sort of plasma when you look at the blood, which is just interesting. And now, so we've done all this talking about how we synthesize fatty acids, how we store them, and how we deliver them to the cells. So finally, if we want to use these fatty acids that we're delivering to the cells, how do we do that? So let's just remind ourselves how fatty acids get into the cells. Like I said before, their pKa is lower than physiologic pH, so they're going to be mostly charged in the blood, and they need transporters. Once they come in, they form this thioester linkage with CoA, converting the fatty acid into acyl-CoA, which traps it inside the cell, kind of like phosphorylating glucose to keep it in. And then, in order to be oxidized, they have to get into the mitochondria, which again needs a transporter. Nothing gets into the mitochondria without a transporter. Well, almost nothing. So how does acyl-CoA get into the mitochondria? Because there actually is no acyl-CoA transporter, spoiler alert. So what happens is the linkage to CoA, which we just made, by the way, gets broken, and the long fatty acid gets transferred to carnitine, which is another molecule, and in this form it can get across the membrane. Then once inside the mitochondrial matrix, the carnitine is discarded, the CoA gets reattached, and we can proceed. So actually, just for the sake of completeness, point of fact, fatty acids that are shorter than 12 carbons can get across the membrane without carnitine, but we're not really going to dwell on that, but just so you know. All right, so now we've finally gotten our free fatty acids coupled to CoA in the mitochondria. What do we do with them? So this fatty acid is going to be oxidized in a process we call beta oxidation, which is basically a 4-step process which peels off 2 carbons at a time. And every step releases acetyl groups, produces NADH, FADH2. And at the end, you're left with an acetyl-CoA which can enter the TCA cycle. And these 4 steps that I talked about repeat for every pair of carbons. So with every pair of carbons, you're generating an acetyl group, you're generating NADH and FADH2. And this is what makes the energetic bookkeeping of beta oxidation so exciting, because beta oxidation is going to give you NADH and FADH2 during the process, and it also gives you an acetyl-CoA which feeds into the TCA cycle. So when all is said and done, when the dust settles, a 16-carbon fatty acid will yield about 130 ATP. So when I said this was an energy-rich molecule, I was not lying to you. This is a very energy-rich molecule. This process liberates a lot of ATP. And this is for a fully saturated fat. For unsaturated fats, they're oxidized similarly, although they require auxiliary enzymes. But you do miss out on the first step, which involves forming a double bond because the double bond is already there. So you do get less energy because this is already a partially oxidized molecule. You don't get quite as many as you would with a totally saturated fat, but unsaturated fats produce plenty of ATP as well. Make no mistake. And just to reiterate also something we said before, we can metabolize odd-chain fatty acids. Although we can't make them, we get them from dietary sources, and odd-chain fatty acids from the diet undergo beta oxidation exactly the same way as others. But at the end, instead of being left with acetyl-CoA, you're left with propionyl-CoA, which is fine. It just gets converted to succinyl-CoA by vitamin B12 and business as usual. Now, very long-chain fatty acids, which are defined as fatty acids longer than 22 carbons, can't be oxidized by the regular beta oxidation. They're too long. So they actually have to be processed in peroxisomes. So they have special uptake transporters which take them into the peroxisomes. And if you have a defect in the uptake transporter, this can result in a disorder in which these very long-chain fatty acids accumulate in cells, and we'll see neurological effects from this. Now, just to sort of close the loop and talk about this in the setting of whole body metabolism. In the fasted state, this is the major time of use for fatty acids in all cells except the brain. The brain cannot use fatty acids, at least as far as we know currently. So the brain continues to use glucose, and that's why we have to preserve the glucose for the brain and use fatty acids and other energy sources in other cells. Acetyl-CoA is a very important energy source. Number one, because it can feed the TCA cycle. And although it can't be made back into glucose by humans, other organisms can do that, buildup of acetyl-CoA is going to allosterically drive gluconeogenesis. We talked about this before, but acetyl-CoA is produced in the mitochondria, and buildup of acetyl-CoA is going to inhibit pyruvate dehydrogenase, which is the enzyme that makes acetyl-CoA by allosteric inhibition. And it's also going to actually activate a kinase which phosphorylates pyruvate dehydrogenase and further downregulates its activity. So buildup of this acetyl-CoA upregulates the activity of pyruvate carboxylase, which drives oxaloacetate production, allowing gluconeogenesis. One more thing that we can do from acetyl-CoA, remember I told you in the beginning acetyl-CoA was going to be important? Acetyl-CoA is very important. Another thing we can do is we can make ketone bodies, which can then be transported to the brain where they're converted back to acetyl-CoA and they can enter the brain's TCA cycle. And they're used as a sort of like cheap knockoff glucose. It's not as good as glucose, the brain doesn't love it, but the brain can function on ketone bodies, which is important because we want our brains to function. So ketone bodies are made in the mitochondrial matrix of the liver. Acetyl-CoA gets converted via HMG-CoA to acetoacetate, which then produces beta-hydroxybutyrate, which is a ketone body, and acetone, which is also a ketone body. And you may recognize the name of acetone. If you've ever heard anyone describe fruity breath of someone who's in ketoacidosis, it could be someone with diabetes, it could be someone who's had a lot of alcohol. That's what you're smelling. You're smelling aerosolization of acetone, and that gives that like sort of fruity breath that's characteristic of ketosis. Now, I kind of mentioned it without really mentioning it, but the potential risk of producing these ketone bodies, these keto acids, is that, as one might infer from the name, they are acidic. So you run the risk of getting ketoacidosis of the blood, which is not good for you. You never want your blood to be hyperacidic. And the ketone bodies that are made in the liver are always exported. The liver actually can't use its own ketone bodies, but pretty much all other cells can use the ketone bodies produced by the liver, except for the red blood cells because they don't have mitochondria, so they don't have a TCA cycle, and this whole thing is not really relevant to them. But mainly, we use ketone bodies to feed the brain because the brain can't metabolize fatty acids as far as we know. And just one final point about regulation of beta oxidation is that malonyl-CoA is a regulator of beta oxidation. Remember that malonyl-CoA is needed for fatty acid synthesis, so it follows that if we're synthesizing fatty acids, we don't want to also be doing beta oxidation at the same time, because then you're just going to be breaking down these fatty acids that you just built, and we're going to be treadmilling. We don't like that. So malonyl-CoA blocks carnitine acyl transferase, which blocks beta oxidation because the acyls can't get put onto carnitine, and therefore they can't get into the mitochondria. They're stuck out in the cytoplasm, and they can't undergo beta oxidation, can't be broken down. Okay, there we go. We talked about synthesis of fatty acids, how we store them as triacylglycerides, and then how we break them down via beta oxidation and synthesize ketone bodies in times of metabolic need. Next time we'll talk about an important piece that I sort of deliberately skipped because it's its own thing in a lot of ways, cholesterol. So we'll discuss cholesterol synthesis, some of the consequences of cholesterol, and then we'll sort of give a big picture lipid metabolism wrap up. Thank you all for listening and for sticking with me. Feel free to reach out at any time to me, medtogether26@gmail.com, with comments or questions, and I'll catch you all in the next episode.